it's a wonderful pleasure and a humbling experience to introduce Trey. He did his undergraduate studies at MIT and then did his PhD with one of the, in my perspective at least, is one of the inventors of systems biology, Leroy Hood. They developed a lot of concepts together, and then Trey, over the last 20 years, really made remarkable contributions to the field of systems biology as well as network biology, is one of the top 1% most cited persons in the field. And so he's been pioneering a lot of concepts, and I think he's going to tell us about some of the new developments. Oh, and developed a lot of tools that all of us are using. Many of you will know it, but maybe not everyone. But Trey is one of the contributors, inventors of Cytoscape, which is something that everybody is really familiar here. And that's only one glimpse of the impact that Trey's having had on the field. So with that, we are very much looking forward to your presentation and your new.
Well, thanks very much, Pascal. Is my mic on? Is mic on? You can hear me? Good. So I hope I can live up to that very nice introduction. Thanks for the invitation, and to the other organizers. I will second what I think Chad said on the first or second day. This is my favorite conference, and it's every 2 years, so it's not very often, but it's nice to be back. So my talk today is going to be organized in 2 parts. Very simple, structure and function, of networks, of course. And the first part on structure, where's David Hill? I saw him earlier. I think he might have left. Oh, damn. Oh, damn. OK, well, Fritz is here. So anyway, so for the structural part, I felt like this was well motivated by David Hill's quote yesterday. It was something, it was sort of a commandment. It was like, thou shalt not attempt to gaze directly at the entire hairball ever. You know, these things, in his opinion, were not useful to look at in their entirety, and I mostly agree with him. The point simply being, I think his point was, all of these interactions never occur together at the same time or in the same contexts, and so what you really want to do is you want to is that David coming in now? Is to pull out pieces of this that are relevant to your particular context. That was his point. David, I'm embarrassing you. So anyway, the second part of the talk is about function. And, and I should say, so in this talk, I will attempt to gaze directly at the hairball. but I will do it by transforming that hairball into something that I think, or will argue at least, is a simpler representation and maybe a more biologically accurate representation of what's going on inside of a cell. Okay, so that's going to be the sort of idea. How do we transform this into models of cells? And then in the second part on function, I thought it was well motivated by Fritz's first lecture, his keynote, at the beginning of the conference, where he showed a lot of funnels. And so I'm also going to show a lot of funnels in the sort of last third of my talk as a way of closing out the conference. But the idea here, if you want to just sleep for the next 45 minutes or go leave early or whatever, is very simple. I'm going to try to take this information, condense it into funnels, okay, that connect genotype to these beautiful phenotypic rainbows. Okay, and you already heard from Pascal. Just to quickly acknowledge the software that underlies a lot of what I'll be talking about. I think for this audience, I can skip Cytoscape. But I do want to call your attention to a sort of newer development out of the Cytoscape group, headed by Dexter Pratt, who's here at the conference. So Dexter, maybe you can raise your hand. But half of you have already talked to Dexter at this conference, if not more. But just to quickly say what we're trying to do with Index. So the idea is a lot of people use Cytoscape to either visualize networks or to compute on networks through things like apps. But a need we saw a couple of years ago was a way to share networks, very analogous to how Google Drive and Google Docs are revolutionizing the way people manipulate spreadsheets and text and things like that. But here the idea is to do the same with networks. And so you can log into Index, you can create your account, you can upload your networks. They're not visible to anyone else besides yourself at first, but then you can create groups of collaborators and share your networks with those collaborators. Ultimately, when you publish, you can flip a switch and it becomes public. As of Cytoscape version 3.6, I think we're now up to 3.8. So it's been a couple of versions now that index can be searched from Cytoscape. So any public content in index or any content that you have permissions to view beyond that public content can directly be downloaded into Cytoscape. But as important, your content can be from Cytoscape uploaded into index. And so we had thought about making this an automatic feature of Cytoscape, but that's a little invasive. So we haven't done that yet. It's still an elective thing. You have to click a button. But please do check out index, especially when you publish, as a way of getting your networks out there. And of course, we've made agreements with journals and are listed in their sort of instructions for how to distribute different types of data. That doesn't mean people necessarily are using this as much as this. So I'm really trying to sort of advertise the utility of index. OK. So now structure. The argument I'm going to make, and I think we've seen this argument made before at this conference, but maybe just not this directly, okay, is that we often in network biology think about these hairballs as the end product of some pipeline. They are, in fact, the beginning of a cell biology pipeline, is the argument. And this argument follows very closely the way structural biology works. And so if you think about the way that works in terms of individual protein structures, you gather some data, whether it's cryo-EM images or X-ray crystallographic images, and there's now relatively well-established pipelines. Of course, research is going on all the time for how you then push a button, and after analysis of millions or bazillions of these types of data, you can get these exquisite protein 3D structures. I think we should start thinking about networks as the raw data, much like these images or these images, and much less like the models themselves. And the question is, how far can we push these raw pairwise distances between proteins? Because that's what basically any kind of network really is fundamentally, is a similarity in some space, functional space or structural space, between two proteins or peptides or genes. And that's very similar to some of the sort of distance constraints you establish in this kind of analysis. In structural proteomics, pairwise distances there are between atoms or amino acids, and so on and so forth. And so it's all sort of pairwise constraints over here. The question is, what are the underlying cell biological structures and ultimately functions that are really revealed and embedded within those constraints? And what should these models look like is a huge, huge open question. So here's the systems biology pipeline we've been developing over the past 5 or so years for how we think you can start with networks as the beginning, not the endpoint of your analysis, and begin to develop those into hierarchical models of cell biology over here. And of course, this is systems biology. So you then want to have this iterative loop where you then use the current state of the hierarchical model to follow back and design new experiments of different kinds of interactions. So you initialize with some data you might have generated in your lab. Of course, you also want to absorb, as I think many people at this conference have shown, all of the public data that are relevant for networks. We, in our process, integrate those into a single data-derived gene similarity or protein-protein association network, fed by all of these features. I'll show that in a second. And given this unified network, incorporating lots of network data appropriately, we then start to detect protein communities in this network in a hierarchical way to build out these structures that look a lot like the gene ontology. And in fact, you can show that in yeast, at least, you can reconstruct most of the yeast SLIM gene ontology of cell component and biological process. But it's now entirely protein network derived. It's a data-derived structure as opposed to a literature-derived structure. And you can compare it to the literature-derived structure to name things and learn what's novel in your structure compared to what's known. So that's the systems biology pipeline. And I really just jumped. I would never at a different conference just jump in with this figure, but I think everyone here is much more on the same page. So now just to quickly go through these different steps. For that first data integration step, there's nothing original in our hands at all. We follow these sort of data integrative approaches pioneered initially by Ed Marcotte. I think Mark Gerstein had a paper around the same time as this one. more than 15 years ago now. And so the idea of Lee et al. Here, and again, many others that have followed, is to take as many network lines of evidence as you can. So here's high throughput protein-protein interactions here, here's synthetic lethal genetic interactions here, and so on and so forth. Integrate them into a one integrated score. They used a different mathematical formalism than we do. We use a random forest here. And so we take all these different features for a protein pair, and we decide how to weight those features essentially by a random forest trained against some target model. And here's where you can argue what the best way to do that is. We think you do have to supervise this, otherwise you don't know how to weight these different lines of evidence. That's the problem. I'd love not to supervise this, but I don't know how not to supervise it. So what we do is, in the example I'm going to show for Autophagy, which we published a year and a half or so ago now, we just took the gene ontology autophagy branch, essentially, and used that as our target for deciding what types of data were the most informative. And it's interesting right away. So in terms of what's most informative for capturing semantic similarities in the autophagy branch of the Go, and this is now for Saccharomyces, I should specify that as well, here are the top 3 sort of most popular kinds of interactions people like to draw. Protein-protein interactions, I think here we grouped both yeast-2 hybrid and APMS type approaches. Genetic interactions like the Costanzo type of interactions. And the third big one, co-expression, because there's so much expression data available always, all the time. And people, of course, will draw co-expression networks out of it. But look what happens. Look at the difference in these networks. Here as you now, so you start, I should describe the y-axis. It's a little cryptic. This is your ability to reconstruct the Go branch of autophagy through semantic similarities between genes. So how well do you match those? This x-axis removes studies from this big integrative approach over here, okay? And whether the study, a paper that is the right unit to remove, we can argue about. That's what we did. It turns out it doesn't really matter. You can remove interaction by interaction as well, and the qualitative results are similar. But basically, surprisingly, you can remove every gene expression experiment from this analysis, and your ability to reconstruct Go doesn't change, okay? However, if you start to remove protein-protein interactions or genetic interactions, and these are genetic interaction correlations, I should specify here as well, that ability does erode, and you can argue which is better. In our case, we moved forward by generating more genetic interaction data because the slope was greater here, although if you notice, there's more protein-protein interaction studies. And so maybe that's contributing to why this trade-off here. But at any rate, it was clear that removing genetic interactions was deleterious to the model, and so we generated more of them. Specific now for autophagy. And so this is, again, I think why starting with a data set and having a target model one wishes to generate is very important, because of course most, if not all, but most of the interaction data we've talked about at this conference are general, okay? They're general reference interactomes. And here we're generating a specific targeted interactome under different conditions that are relevant to the pathway under study. And so here, if you want to activate autophagy, there's a whole bunch of ways of doing it, which of course aren't exactly the same, but here we tried 2 of them. One is to target TOR through rapamycin, which is a well-known stimulant of autophagy pathways, and the other well-known one is to starve cells. And so under rapamycin or starvation or untreated conditions, we could generate these SGA profiles a la Costanzo here, and we targeted essentially every known autophagy gene at the time on the queries versus the genome, essentially. So it's sort of 50 or so versus all. I forget the exact number, but that gives you the idea. And there's 4,500 or 5,000 genes here in the columns. And then in addition to looking at the raw SGA scores in each of those conditions, we also did this subtractive analysis looking for differential interaction scores. That is to say, interactions that appear under rapamycin but were conspicuously absent from untreated. Those get higher weight in those difference interactomes than just an interaction that's always very strong across conditions. And so those were all features that went in over here. And now we integrated them just like I showed via that Edmarkadian approach to get this single gene similarity network, which, of course, as I think all of us know, you can equally view as an adjacency matrix. And so just to kind of give you an intuition for how this process works, of course, you can do this genome-wide, but I'm now going to just focus on a subset of the autophagy genes in yeast. Not surprisingly, many autophagy genes start with ATG. And RF1 at the top here and BCK1 at the bottom are really thrown in just to provide contrast. They're unrelated proteins thrown in to provide contrast in this matrix. So the more red a cell in this matrix, the higher that integrated score. And so then for the high ones, if you want to know why it was high, you have to go back a step in the analysis and ask, well, is that because of a protein-protein interaction measured with yeast-2 hybrid? Is it because of a synthetic lethal interaction or a synthetic lethal correlation? Or is it co-expression, or is it something else? Or is it a combination of multiple lines of evidence? And in fact, that's typically what you find for your highest scores. As one might expect, you're combining multiple lines of evidence measured in multiple interactomes for that particular protein pair. So the eye is very good at looking at these heat maps, I think especially the eyes in this audience, and one can immediately see structure. So we see here ATG and 10. Of course, the diagonal is by default red. Everything's similar to itself. But then we see ATG 10 and 27 form a cluster. But look, 10 forms another cluster with 12 and 7. And look, 7 has the same pleiotropy. It forms a cluster with 3 as well as 10 and 12, and so on and so forth. And then if you put on fuzzy goggles, you can now look at a higher scale here and see sort of a weaker stringency, but still a significant association among all of the proteins in the center here. So the kind of clustering one wants to do to this data is not your typical binary tree construction or agglomerative clustering like 99.9% of the clustering algorithms out there are. Why are 99.9% of these clustering algorithms binary tree constructors? Because when Darwin drew the original phylogeny, it was a binary tree. So when we construct sequence trees and species trees, those are appropriate algorithms. Those are absolutely not appropriate algorithms, building dendrograms for cell structure. Okay, so you want a suite of additional or alternative algorithms I'll get to in just a second. But here's the output of our algorithm here. And so here for ATG10, you can recognize that it's a member of 2 parent complexes or components, as is ATG7. You can take all of these at the higher level, all of these proteins, and place them into a component above that, which nests above, or I should say ATG12 conjugation system and 8 activating complex nest below this higher order machine that you're representing here. So that's the structure of this hierarchy. And then how do you get the names on these things? Well, if you recall in our systems biology circle diagram, there was an alignment step to go. That's the one time we use go information, is just to name components that we found. We don't use it to inject components into this. Okay, so core machinery of autophagy and ATG 113 complex was in go, had a good alignment against that ontology. I should also mention we're using a bona fide ontology alignment, not go enrichment, but you can ask me about that later if you care. The red circled ones were not in go, but were very clear findings in the literature. So they weren't novel discoveries, they just weren't in go. So we submitted those to the go editors, and now they're in. Okay, unfortunately, Go has no way of tracking who submitted what, and so there's no credit given to us for having done that, but you can verify that you can now go find those things. Okay, this last one over here, 183, is neither in Go nor is it in the literature, yet there's a strong PPI and genetic interaction correlation underlying that red dot there, or that red cell there. So ATG 10 and 27 are probably doing something in a complex that is unknown. So that's how you point at novel discoveries using this framework. Now, just a technical point about how you construct that hierarchy. I gave you kind of the qualitative understanding of it, I hope. And I pointed out why your normal phylogeny constructing agglomerative clustering algorithms aren't sufficient. Due to pleiotropy and just simply the non-binary nature of protein complexes. More than 2 things can assemble into a protein complex. So there's been a lot of work in a parallel class of algorithms that are being called network community detection algorithms. Here are some of the more famous ones. published over the years. This is a more recent one called the InfoMap approach. Louvain clustering is this one, or is a successor to this one. Something called clique percolation is over here. But it turns out that there's all sorts of problems with these, what look like, when you read the paper, useful algorithms. In my time here, I don't have time to get into it. I'd love to talk to you guys about it after. And for the purposes of our stuff, we went back to this 1974 paper on hierarchical clique detection. We fuzzified the definition of clique, so it doesn't have to be a perfect clique. But the idea is very, very simple. Okay, so now that we have a weighted network due to integration of all this evidence, okay, some of the individual features are weighted, of course, but certainly the integrated score is. Let's just take a toy example of 8 proteins. This is the actual hierarchy of machines encoded by those 8 proteins. And so let's say this is your similarity matrix, your protein-protein interaction association score, if you like. And so A and B have a relatively high score, C and D have a relative high score, whereas A and H have a 0, okay? Because they're far apart in this structure. So now we can think about this thing as really just a single stringency. And you can start with stringency at the maximum so that no interactions show up at the network, and successively lower that stringency dial all the way until all interactions are present. All protein-protein associations, I should call them here, scored associations. And as you lower that stringency, cliques will appear. And so here's, at the very, first step, you get an edge between C and D and E and F, and those become 2 protein cliques. And those are recorded in your hierarchical construction algorithm. And then you keep adding edges as stringency drops, and those once small cliques start to nest with inside bigger and bigger cliques. So that's sort of the simple idea that we're using here. The problem with this idea, it's very good at reconstructing Go, it turns out, from existing interactomics data. It's extraordinarily inefficient. So we simply can't run this thing on more than about 1,000 by 1,000 matrix in under like a month. And so we can talk about that. So we need better algorithms for finding these communities in large networks, for sure. And then what do you do, of course, once just to kind of show you how you start to explore this. I don't have the data for this one yet, but I do have of course, there's many of these orphaned protein complexes and supercomplexes in these hierarchies that are just left with numbers, not names. And so you start to follow up on them. And so here's one example. Of course, it's a short, 45 minutes is still a short talk. So I'm just going to not show you all the data behind this one. But so there was a complex that was known within autophagy. And here the Go name was something like the pre-aminopeptidase 1 aggregate processing unit. And some of these proteins, like ATG19, were already known to be in this unit, but there were other proteins that were not known to be in this unit, like ATG27 and 26. And so this is just showing you that only when you add rapamycin, maybe why it was missed before with imaging, when you add rapamycin, you get alignment between these sort of punctae that are used here to image these 2 proteins in the cell. And of course, you then do the functional follow-up to that to show these things have similar functional impacts when deleted, and so on and so forth. So that just gives you an idea about how you explore these hierarchies. I don't think that's a surprise to anyone in this room. Of course, autophagy was just an example, and so you can apply this to the entire interactome of a species, like I said. So I said 1,000 by 1,000. In yeast, we managed to power through the 4,000 or 5,000 by 5,000 matrix with this algorithm and get this beautiful flower published back now 5-6 years ago. In humans, we can't. Okay, so we have a sort of staged approach, and it's very unsatisfying, although it may be good enough to get interesting biological results. But let's talk about that. So anyway, so here's the root of the hierarchy. The names here come from alignment to the Go cell component branch. So you can see here there's a mitochondrial part you're discovering and a lot of structure beneath it. There's a membrane part you're discovering and a lot of structure beneath it, which isn't named. So it just this one glance shows you where all the known biology actually is in cell biology. It's all down here in the intracellular component, where you do barely name many of the sort of substructures the interactomics reveal. So just to keep moving, though, to help people build these, so for a long time, this was just something we had to sort of wire bespoke scripts in our lab up to do. But Mike Yu, who was at the conference and I think had to leave last night, but he was here the whole time otherwise, as his last paper in the lab, he's now an independent investigator in Chicago, but as his last paper in the lab, he published this very nice thing he calls the data-driven Ontology Toolkit, which is a Python environment, essentially a set of Python programs that will take your interactomics data, perform this kind of community detection, structure the hierarchies, put them in index, align them against Go to name the parts of those hierarchies you can name. And then with Keiichiro Ono in the lab, who's part of the Cytoscape team, he also developed alternative ways that are part of this DDOT toolkit of viewing these hierarchies. So just to kind of demo that here for the first time, I've been showing these, and we all show hierarchies like this in terms of trees. I should say, due to pleiotropy, it's not a tree. And so here, and in the previous, and some of the previous plots I showed, we've cut some members or some of those pleiotropic edges in the hierarchy just to be able to lay it out as a tree. Otherwise, it's a hairball, okay? That DAG is a hairball. But this alternative view on the right is interesting. So it's the same exact hierarchy here shown of a DNA repair structure we're in the process of building. But in the circle packing diagram, this is what the InfoViz community calls these diagrams, circle packing diagram, a node or a machine in the hierarchy, a system, okay, I'll call them systems henceforth, is a circle. Okay, so the yellow circle here is DNA repair. And the first layer down in that scale, beneath that system, is shown as circles inside of the larger one. What you don't see here, because they're sort of limiting your view, your depth is set at one layer down that you can see. And so if you were to go and click on, say, recombinase activity, that would then become the yellow circle and reveal a ton of structure beneath it. And you can continue to navigate upwards and downwards in these circle packing diagrams called HiView. So right now it's a standalone website, but very shortly will be integrated into the main Cytoscape application. Okay, so that is the first part of the talk on structure. And it was a flyby of the sort of pipeline we're using for a variety of pathways, and we think is fairly general for how you can turn interactomes into these funnels. Okay, so now let's talk about where to go from there. And the point, the major point that I think was already made by Fritz, but I'll make it again, is that these funnels now functionally end up being extremely convenient for translating, or at least thinking conceptually about how to translate genotype to phenotype. So why is that? So first of all, just let me clarify that this funnel is now an alternative conceptual representation of those hierarchies I was just building. You could also imagine the gene ontology as a literature-curated hierarchy that could equally be represented or conceptually thought of as these funnels. And whether it's the data-driven hierarchy of systems or it's the literature-curated hierarchy of systems, starting at the top here, you put proteins into small protein complexes, which assemble themselves into larger, call them supercomplexes. Those supercomplexes participate under the banners of things like signaling networks and different metabolic processes. Those are, of course, in organelles and cells. And of course, the structure doesn't stop there. Now, the gene ontology does, okay, and all of our hierarchies currently do stop at the cell level. But you can also think about single-cell sequencing experiments as essentially revealing the structure of tissues, of all the cell types within tissues. And so maybe, and in fact, the last talk we just heard, I think, had begun to do some of that. And so why stop at the cell level just because Go stops at the cell level? One, I think, in a seamless way, will be able to go above the cell level. And one will also be able to go below the gene level. I realize I'm pointing up when I say below due to the orientation of this thing, but I hope you guys know what I mean. Go essentially starts at genes, but of course you can, and people do all the time, factor genes into domains and eventually amino acids. Okay, so that's this hierarchy that I think all of us are essentially estimating various parts of and versions of and contexts of when we present our interactomics data. And so given that, What about genotype-phenotype? Well, genotype is really states on the very, very, in this case, upper end, but really the lowest physical scale of this model. And phenotype, depending on which phenotypes you're talking about, are at various scales above that in the model. So if you're talking about patient phenotypes, of course, that's at a meter scale up here at individuals. If you're talking about cell-based functional readouts and those being phenotypes, those are phenotypes on objects that are 10 microns, if a human cell is about 10 microns. But for all of these, when we try to go directly between genotype and phenotype, we are trying to bridge an enormous physical scale gap. Okay, because what's the scale? So if the scale of your cell assay or your cell is 10 microns, the scale of your patient phenotype is a meter. What's the scale of the genome? What's the scale of a mutation in DNA? It's a Q&A. And I'll take an angstrom, a nanometer, 10 to the minus 9, 10 to the minus 10. So is it any surprise that genotype-phenotype association is hard when you're trying to bridge essentially 9 to 10 orders of magnitude of physical scale when viewed in that way? And networks provide intermediate detail. And in fact, as I've shown, it's turning out that, in fact, it's not just one layer of scale these networks can inform. It's multiple layers of scale in between the 10 to the minus 9 or minus 10 and the 1 meter scale. So it's a very nice concept. So how does one begin to explore that? And certainly, there's been a ton of beautiful work, much of it presented at this, represented already at this conference, using flat networks in here as a way of interpreting genes underlying disease. That was the session just before us, for instance. So here's the paper we had about 10 months ago, or whatever this was, that was a first rough pass, still quite conceptual, but at least now putting data where our mouth is, for how we might use these hierarchies in machine learning systems that translate genotype to phenotype. And so I've already talked about this, and that's this paper here by Jinzhu Ma, Mike Yu, and Samson Fong in my group, at least at the time of that study. I've already talked about this first arrow here for how the first phase of this is to build the structure from network data. I'm going to stay in yeast for this phase of the talk. In fact, my entire talk, this is the first talk I've given in a year that's only yeast, but that's the way it's going to be. So I've already talked about that. So now what about this vertical axis? That's the horizontal axis. What about this vertical axis? And I should say it's yeast, so that really should say yeast. And this should say, as you'll see, and just as the next slide, you'll see this, that will be yeast growth phenotypes, okay? But the concept is really now we can build these structures in yeast. How do we functionalize them in structure-guided machine learning, as you'll see, deep learning environments? And the last talk, again, was a good précis or foreshadowing, rather, of what I'm going to show you now. So we call this thing DCell for deep cell or something like that. And so the reason, by the way, to stay in yeast here is because not only do we have the best protein interactomes, the best genetic interactomes, well, I should say we also have the best genetic interactomes. And if you think about the protein interactome got used here to build this structure, the genetic interactome is essentially a bunch of genotype-phenotype relationships. And so we don't put that into the structure part, although you could. We reserve that for the function learning part. And so, as again, we've talked about at this conference, and I think half of us know extremely well and have been on some of these papers, we used the Costanzo 2016 Saccharomyces data set among several others, but this is the real driver. What does that data set do from a genotype-phenotype perspective? It's thought of, I think, as a generator of synthetic lethals. Here, I'm not going to worry about calling anything a synthetic lethal. I'm just going to use the raw genotype-phenotype information each of those experiments provides. So the idea is Costanzo has deleted, to a first approximation, every pair of yeast genes and measured a growth phenotype in response to that deletion. So how large is that data set? Well, if we say there's 5,000 yeast genes that they could delete, there are 5,000 squared divided by 2, or 12.5 million pairs of yeast genes that one could delete, and therefore 12 million or so genotypes. Okay, so that's far larger than any patient genotype data set that one can now access, certainly. And so again, yeast is, I think, the right model in which to pioneer and explore some of these ideas. Of course, one also has in this, 12 million is largely dominated by these double these double gene deletions. But of course, there's single gene deletions one has in here, and there's a few triple gene deletions we can put in here too that have so far been generated by, again, some of the labs in this room. Which is another feature of this. We're not restricted to pairwise analysis of anything. Okay, any genotype, no matter how, Fritz can put his green monster strain in here, and we can get an answer. Whether it's a good answer or not, I have no idea. We should do it, right? So anyway, that's the input data for training genotype to phenotype, okay, because I have 12.5 million examples of growth paired with those. And I've already told you how to get this structure. Just to quantify some information about that structure, that flower I showed has about 3,500 cell subsystems in it, if I just count across that hierarchy. So I'm not counting genes and proteins, I'm counting the higher order subsystems that are part of that hierarchy. So it's big. It turns out to be about 12 layers deep, which we thought was quite coincidentally interesting because around the same time as we started this project, a couple years before this publication, of course, the AlphaGo had just been, this deep learner that beat the Go champion had just been announced. And that was also about 12 layers deep and has a few more neurons than this, but it's a comparable size of what we're about to build, actually. We're going to build an AlphaGo-sized thing and as deep thing. So how do we now, I keep skirting around this notion of neural networks, so how do we actually assign neural networks into this structure? And it's very simple. We did the most straightforward thing, even if it maybe not be the wisest thing, okay? So we took each system, like DNA repair, and we assigned that system a bank of neurons. Why a bank, not one neuron? Because one neuron basically is a binary output of a 1 or a 0. And what you're essentially doing here when you assign neurons to systems is you're ascribing a state to those systems. You're giving some kind of state to every system in the cell, as well you should. But who are we to judge that DNA repair is so simple as to be off or on? How many PCs does the state of neural network of DNA repair have? We have no idea. So that is probably the most arbitrary part of this model. We had some sort of function for assigning or trying to figure out what the number of appropriate neurons is for a process, but it's just sort of scales with the number of proteins in that process, but not linearly. That said, now here's the interesting constraint. So now if I have a bank of neurons assigned to DNA repair, I only allow the incoming weights to those neurons to be non-zero from the child components of DNA repair in this model. So as we know, the child components of DNA repair this is not an exhaustive list, are things like double-strand break repair, base excision repair, mismatch repair, and so on and so forth. And of course, there's more here. And so the only incoming non-zero weights in this neural network model are from those children. Now, as the model learns how to associate patterns in the input with patterns in the output, it doesn't have to keep all those equally weighted. In fact, that's the whole point. It'll now adjust those weights, zeroing out even some of these, dialing up the strengths of others to learn to best associate a double or any set of deleted genes here in the input to a growth rate down here. So hopefully that makes sense. So let's now go on to the results. And so, of course, they were good enough that we submitted a paper on this, so it's not too surprising when I show you a sort of correlation like this between measurements and predictions of the model. And here 1 is the wild type rate of growth, and 0 would be dead. So that really should say 0, not infinity, but you get the point. And largely, you're doing a not so great job of predicting increases in growth above wild type. Okay, it's pretty much flat. But you're doing a pretty good job of predicting decreases in growth quantitatively with this model. If you care about these kinds of things, this is in 5-fold cross-validation with lots of devils in those details. Okay, so let's talk about them. If you want to give a single number to this correlation scatter plot. That's shown over here at the right. So this DCell visible neural network, we call it, because it's not a black box. It's a cell biological guided deep neural network. That's about a 0.5 correlation, so that's that bar. Now, I'm not going to talk about all the green comparisons to prior work. I'm going to just skip over here to the orange and yellow bars because they're interesting. So if I now take a fully connected deep neural network with the same number of layers, okay, keep in mind, now that I'm allowing all of those connections, I have way more parameters in that model than in DCell, okay? There's no constraints. Same number of layers, okay? Same number of neurons. So it's remarkable that I would have loved it if the biological model had done better. It does not. But I still think it's remarkable that it does no worse, okay? What's equally or even more interesting is if I now constrain these black box neural networks to be approximately, if not exactly, the same number of parameters, okay, on the weights, that's the same number of non-zero weights as we allow in DCell, and we look at many, many essential ways of, so you can essentially think about this orange matched column as randomized permuted versions of the DCell model, okay? It's the same structure, you permute the structure so you have the same number of parameters. And so the decrease in performance there does argue, at least in this case, that the structure of the neural network as determined by cell biology measurements is more optimal than random structures of the same otherwise equivalence, okay? But that's just a comparison against the black box versions. The real reason to do this is, of course, the interpretability, because it's not a black box, and one can now open the hood and start to explain the reasons why the model at least thinks that there are that these genotypes give rise to the phenotypes they do. So here's an example of a genetic interaction, a negative genetic interaction between two genes, PMT1 and IRE1, okay? So that is to say, when you doubly delete those two genes, the cell doesn't grow very quickly at all. To represent, by the way, state in this model, we're using this blue to red color scale going through 0. So here, blue on the gene level just means these genes were deleted, and blue on the cell state level just means the cell growth decreased in response to that. It turns out it does in a surprising way, so it's actually a synthetic lethal, but that's not represented here. What is here is the explanation for, of the model for why you get this surprising phenotype. And so if you look, and it's very simple for how you pull this out, you simply look at the neurons whose states are most perturbed when you delete these genes in silico now, and you see to which subsystems those neurons map. And it turns out in this case it was pretty clear. The subsystems to which those neurons that were most perturbed mapped was ER unfolded protein response and its parent response to endoplasmic reticulum stress, okay? Now, if you recall, you might be thinking, but Trey, I thought you had a whole bank of neurons here and a bank of neurons here. We do. We're showing you PC1 as a way to just show you a color, okay? There's, in fact, a whole state space one can map out if one wants. You can expand this into multiple dimensions. Here, we're just showing you PC1. But the important point is seen with PC1, which is that the model is giving you an explanation as to why, in the simulation, that deletion results in a synthetic lethality with regard to cell growth. And it converges right here in the hierarchy, okay? Now, you could also say, well, Trey, but Go, I would just look at Go, and it would tell me that those are the parent processes of these genes. Well, yes, and it turns out Go has 239 other parent processes of these two genes, and you'd have no way of knowing which one was the relevant one without a functional simulation of this hierarchy. In this case, I'm low on time, as you see, so I did not plan to show you the data. We can actually validate. We can go in with the marker to ER-UPR and validate that its state in a biological experiment is perturbed by that pair of genes and actually many others beneath this point in the hierarchy. So to summarize, I have argued here for my whole talk, essentially, that networks can and should be analyzed hierarchically to reveal biological systems at multiple scales. Really, I think the way to think about these hairballs as you gaze at them directly is really as raw data points to be used for assembly of models. And the question remains, what are those models? How far can we push those models towards accurate cell biology simulations of structure and function? In this case, we got as far, so far, as to show how these hierarchies not only are sort of structurally interesting, but do seed or can be used to seed deep learning systems for translating yeast gene deletions into growth rates. That is suggestive of other things one might do for translating patient genomes, okay, to other kinds of phenotypes. I think the road there is going to be long and hard for a variety of reasons we can talk about, but nonetheless, I think that's an interesting road, okay? Now, I think hopefully it's been not lost on some of you that we're using a lot of your data. So we need many of the data types we discussed at this meeting in separate sessions. Genetic interactions to learn structure or function across the structure. Protein-protein interactions to learn the structure hierarchy itself. And we need lots more of those kinds of data. And so again, we should talk about it, but one way in which this model may help you think about what to do next is really just to think about the goal here. May not be just to generate the map. The goal is beyond that graph. And that might inform exactly how we go about those next experiments. I briefly mentioned that there's a need for new computational methods, essentially at all points of the pipeline I talked about. Certainly, I'm angry at our network community detection algorithms these days. We're making it work, but it's limping along. And despite all of those beautiful Nature papers that have been published about hierarchical communities in large networks, so we can talk about that. And of course, I think we're at the beginning of how we do this biological machine learning thing, where we guide different kinds of machine learning models with these kinds of network data and hierarchies. But it does start to feel like, you know, we in this room are capable of pushing us into a new phase of cell biology. You know, I was talking with Harvey Lodish after his session a couple of days ago, who of course is the author of this famous molecular cell biology textbook. And we said, shouldn't there just be a systems cell biology textbook? And I think that's what we should really aspire to writing one day. So with that, oh yes, and as a postscript, I have 30 seconds left. As a postscript, I wanted, just because it came up at the meeting, to this point here, need data. I didn't at all talk about our story with regard to synthetic lethals, but that was well covered by Traver, Chad, and others in a genetic interaction session a few days ago. Just to point out, because a question did arise about how sensitive these synthetic lethal networks are to context. We did have an interesting result, again, about a year and a half or two ago, where we had, with Prashant Mali's lab at UCSD, who's the real CRISPR wizard here, I should give credit to, we did this double CRISPR approach to look for synthetic lethal interactions among a bunch of cancer genes, largely DNA repair genes, but a sort of actually mix here. It was like a 73 by 73 synthetic lethal interaction map, but importantly, we did it across 3 cell line contexts. And at least in our hands so far, the networks look surprisingly different. So here, the green interactions are the synthetic lethals that were found in both HeLa and A549 cells. There's a third cell line here that's not shown in this panel that one can also examine. But the point here is just to show that most of the synthetic lethal interactions coming out of the screen were private to one of those cell lines or the other. How much of that is instability and noise in the measurements? Of course, we examined that in the paper, and we try to control for it, but I think the field, has to admit that how we control for noise needs to evolve, versus how much of this difference is biological. That I think remains to be seen largely, but it does give one pause that the networks are so different. So with that, that's my lab, and actually with Anne Roxandra still in it probably somewhere. No, she's gone. Oh, never mind. So I'll take any questions. Thank you.
Beautiful. Where do we start?
Yell?
Yeah, it works.
This one's great, thanks. So a thought experiment, if you gave yourself a budget of one non-zero edge that sort of escaped the shackles of your hierarchy, allowed any edge to be anywhere, but you only get one or five, or some limited budget for non-hierarchical edges, where would they be?
Well, it depends how much you buy the Kool-Aid of biology, or cell biology having to be hierarchical. Because if you say it's got to be a hierarchy, then you would argue that edge is going to add a node. It's going to sort of, if you say you connect two processes together, you're going to join two systems or modules under what will now create a higher order system, I think.
I'm just wondering, can you use your model at the end and then extend it a little bit such that your performance increases and discover
right. So I, in general, let me put it a different way, but I think it's the same. It answers your question, or acknowledges your question at least. We were way too rigid in a sequential approach where we first built the structure and then absolutely fixed it, and then built the functional network of neurons inside of that. Could you build them? Yeah, I mean, you're sort of suggesting a phase 3. That was phase 1 and 2. Maybe now iteratively go back. Maybe there's a way you could function and structure build at the same time. And in fact, there's an interesting paper on arXiv right now out of Yuri Leskovich's lab in computer science for trying to do exactly that. It's not in biology, of course. It's in mainstream machine learning, but the field is moving.
So of course, there's a lot of ideas about transfer learning with neural networks and things. And of course, you would like to be able to use this model also to predict conditional phenotypes, or take a genome from an individual, look at the Lutaris variants, and sort of, is that some direction that you're already working on, or is it?
Yeah, so one question is how to deal with condition, or how to deal with context in these models. Just to keep it general, in some cases of context, you can imagine having an extra input and knowing where to wire that. In other cases, it's not so clear. And it's not helped by the fact that the Go database themselves never really figured out context.
Yeah, but what I'm guessing is, so your pre-trained network that's based on simply essentiality or double essentiality or condition.
You can now shift the condition.
Yes, to an extent. Of course, you don't have the large extent that you have for doubles, but there is some singles.
And so one trivial, non-useful, or marginally useful idea is you could have, so I guess down here at the very bottom is a final node that integrates everything, okay? Before making it's the node that actually makes the prediction of growth at the end. You could wire, if you knew nothing about where that context affect, so let's say you heat shock the cells. Should you wire heat shock to HSP components inside of the hierarchy? Maybe. Or should you throw your hands up and just wire it to the end? Make sense? Kind of. So I can think about how to represent those environmental shifts. I can't decide if they're good ideas or not.
Hey, so I'm thinking about the structure part of your talk, and also particularly to do with context, learning the autophagy network. So of course you said that you were collecting data specifically around autophagy, and then you also had a bronze standard for the Go branch around autophagy. So how important was it that you used that autophagy branch of Go, and if you'd used other parts of Go, how much of an effect would that have had on your inference?
That is an excellent question, because I actually don't, I'm very uncomfortable using Go at all in this process. Now, and so I don't know the answer to your question, except for one part of it, which is we did show that if you just train on autophagy, the model predictions are pretty well correlated with if you trained it on the whole Go. And I think one of the reasons why you got that stability was, if you think about it, there's not a lot of parameters here. Now, random forest is a confusing thing, but let's just say it were a linear model, okay? So then in a linear model, you have a single weight for every kind of interaction network or type of interaction network. So yeast-2 hybrid gets a weight. Mass spec-based interactomes get a weight. So it's one parameter per input data type. It's not, so there's like 20 or 30 parameters you're fitting. You're not fitting a million parameters. And that might be the reason why it's not such a bad thing to do. Because of course, what you're really interested in is just because you weight the data in a certain way is no guarantee you're going to recover Go. And of course, it doesn't constrain you from finding lots of new components that aren't in Go.
Yeah, so we've been working on context-specific network inference around EMT.
Yeah.
And we've got a different way to developing what we would, your bronze standard or your gold standard. So it might be interesting to talk a little bit more about that later on. But yeah, it's very interesting, that map inference.
I just have a quick one. I just want to be clear. So bronze is Go. Silver in vitro. No, I'm serious.
Silver is Patron.
So bronze is Go, silver is in vitro, and gold is in vivo. I just don't know where those are terminologies.
I called so I was simply using the word bronze to acknowledge that Go is not perfect by any means. That's just, I mean.
Should we, as a
one
community,
well, but systematically,
agree?
how does one? I mean, the point is you don't know it.
Say it again?
I mean, if you knew it, then you wouldn't have to do the experiment.
No, I understand, but the Go is bronze, silver could be in vitro, and gold could be in vivo. That's my suggestion.
As a validation, absolutely.
But is that a standard? Do people use those?
If you were able to do enough experiments, I mean, you'd have to, But I mean, yes. If you could do enough experiments, what's nice about Go is it gives you some target for essentially, gives you some information about the structures every protein participates in.
Yeah, but I agree it's bronze because it's not, functional data. Yeah. It's not actually experiments. Okay.
Well, and it's highly filtered through Go editors and linguists and really had a very different purpose, if you think about it, than we here in this room have when we're trying to model cell biology.
Hi, really cool talk, interesting stuff. So when you showed your evaluations, you had this matched network, which is your randomly permuted thing, right? So it's doing better than every other black box method. It's not as good as the one with the actual structure.
99 something percent.
Sure.
Right.
But it's not doing as well as the one with. It's actually informed by the gene ontology, but it's still doing really good, right? And that's totally uninterpretable. You can't reverse engineer what's going on. Yeah, the orange box there, right? Like if you got the weights and did your principal component analysis, all that stuff, it would be uninterpretable.
And
right?
actually, that's why exactly. And well, let's talk about that. So the first order effect I think we're seeing here, and this is a little bit of speculation, and the reason I don't talk too much about these green comparisons to very old approaches now is deep learning is very powerful. Throw deep learning at a problem and the predictions get better regardless in many, many people's hands, and the same is true in our hands. So I think really just deep learning, I mean, this is basically even sort of randomized structures. Deep learning is still able to set weights in those fixed structures and learn to associate input patterns with output patterns.
Right. But so my question comes down to how can you know that in the network that was trained with the gene ontology structure in mind, like your DCell VNN, how do you know that that actually is interpretable, given the fact that you can get really good predictions?
How it is not, is or is not interpretable? Is not.
No, how do you know that it is in fact interpretable?
That the hidden nodes represent biology.
Yeah, do hidden nodes represent biology?
Exactly.
That's right.
Think, right?
The answer is no.
But,
and we looked at, so that I can say no. And the reason is, here's the interesting finding. So if you So it turns out there's already some random initialization when you construct these deep networks. So every time you press enter, you do get a slightly different network or a vastly different network. The predictions are always exactly the same in terms of the accuracy. But now, so if we look across different runs, essentially, of building this thing, okay, you can think about it kind of like bootstrapping, okay? And now, so the predictions are almost always the same. Okay, those are stable. What about the structure inside of the black box? It's totally different. So it simply wasn't stable enough for us to do that, or that analysis, if that makes any sense. What I think is going on, again, it's a bit speculative, is there's again many parameters here. In even these models, there's a lot of parameters. And so there's many different neural network configurations that essentially make equally good predictions. What you're doing over here is you're picking the one of those that has evolved.
I guess this question depends. The fact is that how are you sure that the hidden nodes in your biological network actually represents those hidden biological meanings?
Well, that was this argument here. So, and that maybe gets to Gloria's point. So the way we were thinking about what she was calling the gold standard stuff is when you make predictions. So how do I know that the state of ER is really what it says it is? Now I have to go and do the experiment. And so if you read the paper, we did it for this one and one other. The problem I have there is it's not a high throughput thing at that point. So how do I systematically validate the model functionally beyond what's been done is an open question.
But in principle, you could go to all the Constanzo positives, pick them out, put them under the microscope, and look for predicted morphological phenotypes predicted by your algorithm, right?
And maybe if one can, if single-cell RNA-seq ends up working, you could get a RNA-seq profile for every single one of those double and triple gene deletions, and that would really let you then match those states to states of this, Maybe that's how it has to go.
Hey, thank you so much. I have two thoughts I want to share that echo some of the previous questions and that I want to challenge you for the future of this development. The systems biology of the cell can come out of these active hierarchies, but we need names, and we don't have names for what we don't know. And where you find names because it's already in Go, it's fantastic, super helpful because it helps us generate new hypotheses. But you showed, for instance, the entirety of what happens at the membrane in the 2013 Nexograph flower is unnamed. I exaggerate. So can you please work on some way to provide us name? I don't know, text mining, semantic networks, something to help us.
I totally agree, and that would be one route to then generate lots of follow-up projects on these orphan structures.
That would be so invaluable.
And it'd be great to get that pipeline really established, which we've done these one-off experiments, but how do you, what's the next step of that, I think, is
right, when
really
there's no name, there's no assay to validate.
But let me challenge you back.
Oh.
So if you could really get a machine learner working where you did a structural set of experiments to build out these hierarchies, you then maybe simultaneously, as we were talking about with Fritz, are fitting functions into those hierarchies, why do you need to name why do you need to call that the ribosome? The machine knows very well what its function is, and it will have an address in the machine that ends up being the ribosome, but why do you care? You care, of course, when you want to understand things and map it back to what you knew. It won't keep the machine from working, though, not to know.
Right, but I hope you work not for the benefit of the machine, but for the benefit of science.
But the interpretability is gauged.
The interpretability is
knowledge.
key, right?
Well, but it is structurally and functionally simulating
absolutely
biological systems.
Absolutely.
Right?
Yeah.
And so, for instance, let's fast forward. You throw a cancer patient through the cancer version of this. It explains that cancer genotype and maybe some environmental effects once those get incorporated by some paths through this hierarchy. Maybe those paths, none of them traverse unknown terms here, but maybe those paths traverse many unknown systems or an unknown system. At that point, you become very interested in finding out what that thing is doing.
Indeed. That's,
Right.
yeah. And the second thing which is related is, at this meeting, we've heard a lot about non-hierarchical relationships within systems. So hierarchical or crosstalks or relationships that don't directly map directly to such a beautiful hierarchy of structures. Do you have plans, or please make plans, to think about how to incorporate them?
So this is a bit like, I forget.
It's linked to the previous comment.
A previous question.
Yeah.
The way you deal with crosstalk, or the way Go deals with crosstalk, is it connects those two crosstalking pathways under a parent term. It doesn't violate the hierarchy at all. In fact, that's the whole purpose of it.
Oh.
It just means when you see crosstalk between two things, either they're crosstalking because of wherever their ancestral machine is, right? Or there's a lower, more specific ancestral machine that isn't known that's responsible for that. I think it's okay.
Maybe, if they are then interpreted differently by the predictors.
This is all very speculative, I know.
Yeah.
But.
Just from a practical point of view, you've done this first with yeast, where you have like, I don't know, we have 12,000 out of 15,000 predicted interactions. When you do a sensitivity analysis, when does it stop working? How few interactions can you have? And the approach of reconstituting the structure is still functioning and working.
Yeah, that's we did that experiment, and I'm scratching my head to remember what the answer was. But it was it's a great question because it informs how much data one might need in the future for another run of this. So for protein-protein interactions, I don't know, but for the genetic interactions, we did dial it back from 12 million genotype-phenotype pairs to 6 million, 3 million, 1.5 million, and so on. And again, don't quote me on this, but I think it breaks around 1 million.
Okay.
But it's a great, and one should look at that.
So roughly 10% of the search space.
Please don't take that. It is quite late. But
it is late.
Yeah.
Oh, it is past late. Yes. We all thank you for the reminder. So let's all thank Trey first for a very inspiring talk.